stakeholders. Dr. Datta's academic and research training in neurotechnology, motor rehabilitation, clinical neurophysiology, and cerebrovascular medicine provided him with expertise for translational research focused on developing neural interfaces to monitor and activate beneficial neural functions. Over to you, sir, for the talk.
Thank you. Thanks much for the introduction. So I'm currently with the Department of Biomedical Engineering at the University of Buffalo in USA. So in today's talk, I'm going to present some preliminary results from our ongoing work on a phase 0 human brain organoid platform that we are developing for neuroengineering optical theranostics. Now, let me see if I can. Okay, before I go into the details of our work, I would like to kind of introduce and provide some background. So we live in this world where we have electromagnetic radiation. So that goes all the way from non-ionizing radiation, which is at the lower frequency, than the ionizing radiation, which is at the higher frequency. Now we use this, so these are used for different purposes. So we have this radio spectrum that we know that we use for cellular communication, for TV, for radar, for satellite communication, and this is considered to be not harmful. However, recently, depending on, of course, the dose, there are studies that have shown that this non-ionizing radiations can also be harmful to the human tissue. Now, on the other side is the ionizing radiation, and we know that these radiations are harmful. They cause damage and they go deeper in the tissue. However, they have benefits also, because if we can keep the dose below the threshold, threshold of harm, or this SAR, the specific absorption rate, then they can be used to image the body. For example, X-rays and gamma rays are used quite commonly. Now, in between this radio spectrum and the X-rays and gamma rays is this terahertz band, and then there's this infrared visible spectrum and the ultraviolet band. So now, recently, this terahertz band has been being increasingly investigated for not only imaging but also for modulation of proteins. Specifically, in my talk, I'd be looking into, or I'd be presenting this near infrared light and how that can modulate the cellular activity. Okay, so now if you go to the, so now in the next slide, I'm trying to introduce this photobiomodulation, which is where so this is the hypothesis is that the light in the near-infrared spectrum can modulate this this membrane protein, cytochrome C oxidase, and the cytochrome C oxidase is the terminal enzyme in the electron transport chain that produces ATP, and ATP is the energy currency. So dysfunction in the cytochrome C oxidase kind of backs up this ETC chain, electron transport chain, and affects the production of ATP. Now, the hypothesis here is that the near-infrared light around 800 nanometer can modulate the activity of cytochrome C oxidase, which reduces oxygen to water, and this change in redox property will accelerate the electron transfer and the ATP production. So this is one of the hypotheses for how this near-infrared light can be used to modulate mitochondrial function. And also, so this is what we call the redox modulation. So this is in the center, the picture center. I'm showing this different hypothesis for this photobiomodulation. So the redox modulation has this one aspect. The other aspect is this NO release. So the photodissociation of NO can also be facilitated with this light, 10 nanometer light. And that actually can cause vasodilation. So this can be used for vasodilation. But Besides this redox modulation, there is also this ROS, or reactive oxygen species generation, which is kind of considered a negative effect, which includes superoxide generation and photodynamic action, which can be used for cancer cells, but then for healthy tissue, this might be detrimental. And then it also has a heating effect, and the heating effect, not just for the cytochrome C oxidase, but in general, the tissue, the temperature can increase, and that, for example, for neural excitability, can make neurons more excitable, and can affect other aspects of the tissue, besides the cytochrome C oxidase. Now, currently, the mechanism is not well known, but definitely all these mechanisms are involved, most likely involved, and it basically depends on how one pathway interacts with the other pathway, and what's the eventual effect on the cell, in terms of gene expression, in terms of intracellular calcium, NO production, the mitochondrial membrane potential. So these all change, and this is a complex interaction that that is actually under study for quite some time now, and we have some understanding of this that I will present next. So now, before we go into that, just to kind of introduce the clinical application. So this light has to be delivered to brain tissue if you want to modulate the function of the cortical tissue, and that can be done transcranially, as you can see on the picture on the left, figure A. But one can use also an invasive way of delivering to the deeper subcortical structures. Can be done intranasally, can be done intraorally. So these all different ways of delivering the light, and has a different amount of attenuation due to the tissues that are in between. So we did a modeling study where we used a human head model and looked at the drop in the fluence rate, and that's shown on the right. So you can see that there's a sharp drop at the scalp to skull interface, and then at the CSF, cerebrospinal fluid, and about 1 to 5% of the light in the NIR spectral region reaches the gray matter. And this is when you deliver it transcranially. So now what we found is that this 810 nanometer that is commonly used for clinical application has, of course, higher penetration depth than 630 nanometer and 700 nanometer, which have been used also. And this kind of supports this idea that 810 nanometer may reach the gray matter and can modulate the neuroenergetics, or primarily the mitochondrial function there. However, you can also imagine that different people, different individuals will have different skull scalp thickness, cerebrospinal fluid. So there would be a lot of variability in the amount of light that is delivered to the gray matter, so the tissue. So I will go into a little bit more detail on why this is important, but you have to keep in mind that this is kind of like what probably has led to a lot of variability in the clinical studies. So it's not like it's being under investigation for quite some time now. However, in human studies, there have been very few successful human studies, human clinical studies. And this probably is due to this variability in the dose. And our goal, or my goal in my lab, is to kind of use a neuroimaging technique in combination with the stimulation technique to monitor the dose that we deliver to the brain tissue. Now, just to kind of mention that there is this double-blind placebo-controlled trial going on in Alzheimer's by a company in Canada. They have this NeuroPX gamma, which again uses 810 nanometer light at 40 hertz pulse. So that's some introduction to to where we stand in terms of clinical application. Now, before I present our own results, I would like to highlight this extensive work by Dr. Hamlin, who has investigated this dose response. So what I'm showing here, so there are slides by Dr. Hamlin. So on the left, what I'm showing is this intracellular calcium after 810 nanometer stimulation. So the plot so the bar graph at the bottom on the left shows the fluence. So you have the control as the first, and then you have a different fluence of 810 nanometer laser light here. And then what you can see is that there is this biphasic dose response. So it increases, reaches around 3 joule per centimeter square, kind of the peak, and then it then again decreases. And at high dose, it's kind of, comparable to the control, so it doesn't make a difference. Now, if you look at then the mitochondrial membrane potential, also at 810 nanometer, then this also has a biphasic dose response. So you have this control on the left if you look at the bar graph, and then as it increases, you have an increase in the mitochondrial membrane potential, and then it decreases again. So now, but you can see that there's a relation, right? So this reaches peak around 3 joule per centimeter square. The intracellular calcium, mitochondrial membrane potential also reaches a peak around 3. And then in the next slide, we'll look into the ATP levels. So this also has this biphasic dose response. This increases with increase in the fluence and then decreases. And we have around 3, the peak, similar to what we had with intracellular calcium and also mitochondrial membrane potential. Now, when we looked at, so those are the one aspect, but if you look at the ROS production, this has a triphasic dose response. So it increases first up to this 3 joule per centimeter square, then drops, then again increases at higher fluence. So we have to understand this ROS because, like I mentioned, this is kind of a double-edged sword. So at low concentration, this might be beneficial in terms of facilitating gene expressions, beneficial gene expressions. However, at high dose can cause harm to the tissue. Okay, so now I mentioned about this NO level, which can cause vasodilation. So this also has this triphasic dose response. So as it increases, as the fluence increases, you can see with the bar graph at the bottom, it increases first, but at 3, it actually dips. So it has actually the lowest. So the NO has a different response than what we saw with the calcium, with the ATP levels, and with the mitochondrial membrane potential. So it has this dip at 3, and then again it increases. So that's a triphasic dose response. And what we are specifically interested in for the clinical application is that this light can actually somehow ameliorate glutamate excitotoxicity. So it can do away with that. And this is again based on this preliminary prior studies done by Dr. Hamlin, where he showed that. So if you look at this bar graph again on the right, they used 3 joule per centimeter square, which was, as you saw in the prior plots, had this peak for calcium, intercellular calcium, for mitochondrial membrane potential, and for the ATP. So, and then you see as the glutamate concentration increases, the cell viability. This is an MTT assay, which is for the cell viability, the fraction, and that decreases with increase in glutamate concentration. So more glutamate, there's an excitotoxicity, and the cell dies. So there's less viability. But with this light at 810 nanometer, you have an increased viability of the cells. So this is kind of promising because this glutamate excitotoxicity is very common in different neurological disorders, including hypoxia, then epilepsy, and also this kind of is interesting. But the aspect that we want to kind of take home message from all these studies is that the dose is very important. So if we cannot deliver adequate dose, it can also have a negative effect, and of course the benefits also will change depending on the dose. So this is kind of the motivation for the work that we are doing with Brain Organoid platform. Now, this Brain Organoid platform started in collaboration with Dr. Michal Stachowiak. So Dr. Michal Stachowiak has worked in this neurodegenerative diseases, specifically looking into the different pathways, and he started looking into this. He started developing this human organoid model for corticogenesis. So this is during the neurodevelopment, how the cortex develops can be modeled with organoid. So briefly, there's this different zones in the cortex. So we start with this ventricular zone, then there's a subventricular zone, intermediate zone, cortical plate, and this different layers, they develop with the development of the brain. So there are these neurons, migrating neurons that emerge from the ventricular zone, and then they travel up the radial glia fibers to the cortical plate. And this is very important that they travel and integrate. And this younger neurons, as they travel past the older neurons to the surface of the cortical plate, interact with them and creates this circuit that we know with this excitatory neuron and inhibitory neuron. So this integration process is very important, and this is what Dr. Stakowiak found affected in the schizophrenia. So on the right, what you see is the organoid. Then you can see this labeled with this PANU, which is this key somatic nuclear dendritic axonal proteins. And then you also have this proliferating neurons in red by Ki67. And that's actually in the VZ. So if you see in that picture, you have this label VZ, IZ, and CZ. So that's the cortical zone, intermediate zone, and the ventricular zone. And you see this nice red labeling of the proliferating neurons in this VZ and intermediate zone, ventricular zone and intermediate zone. So that's in the control on the left, top left. But if you go into schizophrenia, then you see, so this is at the 2 weeks, you see some of that neural proliferation in red, but it's not that organized. So you see a lot of disorganization of the three zones, in fact, ventricular zone, intermediate zone, as well as cortical zone. Now at 5 weeks, again on the left, you see this nice, in control, you see this nice proliferating neurons in red, but in schizophrenia, you don't see them. So it's all mature neurons, or what's called a neural committed cells. So they are all green here. So this is something that he highlighted, and he mentioned, and he kind of came up with this idea that schizophrenia develops in the first trimester, and we need to look into this more carefully in how this is affecting the function of the brain. So this was kind of the motivation for our brain organoid platform, and then Sneha worked with me and Dr. Stakowiak on developing this instrumentation. So this instrumentation, essentially, we wanted to keep the instrumentation such that it's translatable. So, I mean, it can be translated to human studies. So we wanted to measure it from the surface of the organoid, just like we'll do from the brain. So we have this tetrode array. So if you look in the left, we have this organoid embedded in the matrigel. So matrigel properties, electrical properties, are like similar to skin. So it can kind of consider as this organoid as a brain, and then the matrigel as the kind of a skin covering. And then we embedded this tetrode array in the matrigel. But we didn't actually, I mean, it didn't penetrate the organoids. It was kind of as close to the surface as possible, but primarily in the matrigel. And then from the bottom, we had this optical fibers that delivered light. So the central fiber delivered the light, and then the scattered light was then collected by the surrounding fibers. And that was used for spectroscopy. So for broadband spectroscopy, which was then stored in the computer, then the tetrode collected the electrophysiological data, and this was again, we used an amplifier, then data acquisition to store it again in the laptop or computer. And both the systems were triggered, so they were synchronized. So we could actually monitor, although sampling frequency is different, but then still they actually started at the same time, so we could actually relate in the time. So here specifically what we were interested in is this redox state of the cytochrome C oxidase, like I mentioned earlier. So this is kind of a terminal enzyme of the electrotransport chain, and it plays an important role in the ATP production. So we did, so this is a broadened spectroscopy. So we had, we used light within this visual and infrared spectrum. to image, to monitor the cytochrome C oxidase, and we fitted a model which is explained in detail in this paper. But what mainly you want to keep in mind is this absorption at the 600 nanometer, which plays the biggest role or the most important role in how we measure. So here you can see that the reduced cytochrome C oxidase has a higher absorbance than the oxidized cytochrome C oxidase. So as the light travels through this organoid, if 600 nanometer is absorbed more, then we know that it's in the cytochrome C oxidase in the reduced state, more in the reduced state than the oxidized state. So this is kind of the idea behind this spectroscopy technique for cytochrome C oxidase. And then of course electrophysiology is it will collect the local field potentials as well as some spiking activity depending on how close are those electrodes to the neurons. So if they are very close to the surface, then they will also pick, and we did also see spiking activity in our recordings. Okay, so now what's the method? I mean, the signal processing method so these are like non-invasive recordings. So we have to kind of process them to extract some meaningful information. So one of the aspects that we want to look into is this synaptic inhibition excitation ratio, so EI ratio. And there's this, in the neuroscience field, there's this hypothesis that this balance of EI or excitation inhibition, synaptic inhibition, excitation inhibition facilitates the functioning of neural circuits, and any failure to maintain that balance will lead to a circuit dysfunction. And this has been shown in various neurological disorders, diseases. Now, so this is something which is a synaptic level, and we are trying to measure this non-invisively. So we looked into literature to find what the best way to measure this with our electrophysiological data. And we found this Gao et al. Who have shown that the spectral parameterization of EI ratio. So this is a very nice paper that was published in eLife in 2020, where Gao et al. Showed that this cortex-wide, showed using cortex-wide transcriptonic analysis that there is a direct alignment between the time scales. And I will show you what the time scale is. So time scale of neural activity and the expression of the excitation inhibition related genes, and also the genes specified in the voltage-gated transmembrane ion transporter. This is expected. So voltage-gated transmembrane ion transporters will affect the timescales of the neural activity. However, it was interesting that this was also related to this excitation inhibition-related genes. Now, when I say time scale, so this is essentially this autocorrelation function of the time series. So, for example, for local field potential, and as it so on the brain surface, this has variability, so it changes from areas to areas, and that is actually has this direct alignment with the expression of excitation inhibition genes. Now, what the method that we use in our study is the spectral parameterization. So if you look into the power spectrum of the local field potential, so it has this around 10 hertz oscillatory peak, and then there's this drop, kind of like linear drop. So it's in log scale. Okay, so frequencies in log scale. So if we take out the oscillatory peak and we can then fit a line, which is basically a slope of this drop of power after the oscillatory peak, and this can be related to the EI ratio. So Gao et al. Did simulations in his paper, and he showed that this slope can be related to the EI ratio, and this is what we used in this in our study. So this is essentially our approach for how we measured EI ratio, and this is important because like 1/10 of the gray matter in the brain is occupied by mitochondria, so it plays a very important role in processing of synaptic information. And this whole goal of this work was this translational translation to human studies, because we were using non-invasive ways of recording this, so we could use this in human clinical studies as well. Okay, so now some results, preliminary results. Now, one of the things that we, so this is again a preliminary result that we already have published. So we first look into this healthy neurodevelopment. So how the healthy organoids they develop. So here what you see on the x-axis is this the control organoid at the 23 day, then 2 month and 3 month. Okay, so there's three time points of the neurodevelopment. And what on the y-axis you see is the cytochrome C oxidase signal. So this is, like I mentioned earlier, we use this Broadband spectroscopy, and this is the signal. So, um, so what we found is that with the neurodevelopment, there's this um, decrease in the signal, which is related to an increased energy production. So this is one aspect of this organoid development in healthy. The second is this network function which we estimate, which we measured using the spectral exponent that I mentioned. So what we found is that so what we found is around 2 months there's a lot of variability between organoids. So these are all healthy organoids. However, there was a lot of variability in the 2 months, but at the 23 day and 3 months, if you see there's a decrease in this spectral exponent, which is proportional to the EI ratio, that also decreases. So there's a decrease in EI ratio with maturation, with the development of the organoid. So now, so what we concluded from here, again, this is like not a, I mean, we are still continuing this work, but based on this data, what we can say is that the ox-force energy production increases and the EI ratio decreases. So there's more inhibition, most likely. With the neurodevelopment, with the maturation of the organoid, healthy organoid. So now, when we look at the schizophrenia neurodevelopment, then again, this is a snapshot of the study. So I'm just showing this one organoid, stage of the organoid, which is at the 34 day. So on the right, you can see this orange, which is this schizophrenia organoid at the 34 day. And then on the left are again this control 23 day, 2 months and 3 months. I'm showing in the cytochrome C oxidase signal here. So what we found is that the oxidized state or the redox state of the cytochrome C oxidase is similar to the healthy control at 2 months. So at 34 day, the redox state is similar to what we had in 3 months, at 2 months for the healthy control. So this kind of showed, and we then also now have more data, but the idea is what we found is this, that the schizophrenia has this high oxphos, oxidative phosphorylation function that is attained faster than the healthy control. So somehow the schizophrenia organoids, they are actually kind of this oxidative phosphorylation function is developing faster than the healthy control. So then we looked at the mean spectral exponent. This is again related to the EI ratio. And here again, so like I showed earlier, so there's a lot of variability in control at 2 months, but then 23 day and 3 month, you can see kind of a trend there. And the schizophrenia organoid kind of fits in the middle where, again, it looks like the schizophrenia networks are maturing faster. So the 34 day organoid kind of sits where a healthy control will be at 2 months. And this is kind of a indirect measure. So Dr. Stokoviak has also done histology where, like I showed earlier, we see that in schizophrenia, there's more mature neural committed neurons than progenitor. So there's also this, from histology, we know that this organoids are developing faster. But it was interesting to see kind of that reflected in this mean spectral exponent measure. So that was interesting from this study. Now, one of the things that I wanted to kind of was very interesting, and this is something that maybe modeling can help, or computational modeling can help, is this spectrum of the local field potential. So if you look at, so this is on the left is the healthy control at 60 day. On the right is the schizophrenia at 34 day. And so if you look at the spectrums on x-axis of frequency from 0 to 50 hertz, and if you look at this power spectrum, you see that this alpha oscillatory activity is there at 60 day, but in healthy control, but there is not much oscillatory activity at the gamma band. But in the schizophrenia, so in the schizophrenia, we actually at the 34th day we have the alpha, but we also have this gamma band oscillatory activity. So it's like somehow this is maturing faster. We do see that in the control, but much later. So this early arrival of the oscillatory peak near the gamma band is what we have noticed, and this is actually so this is the preliminary data that we already published, but more data that we have also shows this effect of this event of early arrival of oscillatory peaks. Now, there are a few things that actually we are postulating based on this data. So one of is this gamma band activity is very expensive. So it's energetically expensive. It's a high frequency activity. So the question is whether this early arrival of this gamma band peaks, whether it's actually making the mitochondria go into this OX-force overdrive. So there's this larger requirement of ATP that needs to be satisfied by the mitochondria. And then if, for example, the mitochondria cannot produce or deliver that energy, then whether that can lead to excitotoxic cell death, and which is also has been seen in the histology that Dr. Stakowiak group has performed. So this is kind of the relation that we are trying to make between the maturation of the organoid, the amount of energy that is necessary from mitochondria, and then from work in my lab, we're trying to see if the photobiomodulation can somehow facilitate this energy delivery or the ATP production, and that's can abrogate this excitotoxic cell death. So this is kind of the ongoing work in collaboration with Dr. Stakowiak. So now I want to so this is unpublished. We are working on a publication. So what I wanted to highlight with photomodulation is in the healthy brain organoid. So we did a 10 minutes of photomodulation at 810 nanometer. So a few things we noticed. One is that there was a change in the pH from 7 to 9, which is become more alkaline. Then there's an increase in temperature at 0.2 by 0.6 degrees Celsius, which is again, this is not in bioreactor, so there was no circulation. So this is just the organoid setting in media, but in a bioreactor with circulation, maybe we'll be able to keep it lower than that. This, and then the spectral exponent in the 1 to 40 hertz frequency band decreased. So that kind of shows that the EI ratio decreased. So it's probably more inhibition. But then this also needs to be established with histology to be sure that there's a substrate there that can be linked to this functional data. And the CCO activity, cytochrome C oxidative activity also increased, and the energy production increased with photomodulation, with 10 minutes of photomodulation. So this is the preliminary results, and this is an ongoing study, and we are looking into in schizophrenia organoids now how the photomodulation affects these parameters. Okay, so now for discussion, so this is all the results. Now for the discussion, what is the hypothesis here? Like, so there's a lot of this different results that I showed. Now we are pursuing this hypothesis for our future studies. This preliminary study, kind of pilot study, provided some insight into the into what's in terms of the data. But for now, for the scientific study, we have the hypothesis that in schizophrenia, the OX-force goes in, the oxidative phosphorylation goes in overdrive. And this is supported by this recent paper by Zhang et al. This mitochondrial hyperactivity contributes to the social behavioral impairments. So here the idea is that with an hyperactive mitochondria, you have a reduction in the GABAergic signaling, and that probably leads to this dysfunction in EI ratio. And this is because of this this RLR component of the mitochondria that sequesters the GABA. So this essentially needs to be looked in more carefully with cell culture studies. But it was interesting that we could pick some correlate of that using our approach within our non-invasive imaging approach. So this is the direction in which the kind of the scientific study is going with Dr. Stokowiak. So I'm all interested in photobiomodulation, and the idea is whether we can ameliorate this by increasing the oxphos efficiency of the mitochondria with photobiomodulation. And photobiomodulation not only in the NIR spectrum, so which is the 600 to 850 nanometer, but also we are trying to see if the temperature change can also have an effect, which is from 900 to 1100 nanometers. So there's different spectrum that we can use to kind of modulate the dynamics here. But again, a computational modeling would actually can provide much more insight than just looking at the data and trying to understand what's going on. So this is our current hypothesis for the scientific work. So in summary, I just wanted to highlight the results. So we found that with increasing maturity, there is an increase in cytochrome C oxidase activity, so it's higher energy production. Then the schizophrenia organoid, they were maturing faster, both in terms of cytochrome C oxidase activity and also in terms of EI ratio. And when I say EI ratio, it's kind of difficult to understand from this ratio what's going on, because you can have an increase in I that reduces the EI ratio, but you can also have death. Because of excitotoxicity of the excitatory neurons, and then that will also reduce the ratio. So we need to also look into this histology to relate to how that's connected to the EI ratio that we are measuring with electrophysiology. And then for our ongoing work, this idea of bioenergetics of schizophrenia organoids is our focus, and hypothesis that it's putting the mitochondrial oxphos in overdrive and possibly can be ameliorated with photobiomodulation. So this is, in summary, our current state of this research. And basically, so we are now also going to the genomics part to understand the gene interaction. Okay, so that's it for from me. Now, if you have any question.